Okay, pay attention. First I will tell about immune reactions. Okay, what are immune reactions? To be precise, I am discussing about hypersensitivity reactions. Okay, so how immune system will attack? How the immune system attacks? Okay, so I will show you the four types of attacks. There are type 1, type 2, type 3 and type 4. There are four types of attacks or destruction can be done by immune system. So this is a pathogen, so I am giving pathogen in all the places. Assume this is a pathogen and how this pathogen will be attacked by the immune system. So in type 1, what happens was, there is a cell called eosinophil, and also we have mast cell. 1 second class. Alright, sorry class. Okay, pay attention, where am I? Types of immune attacks. So listen, the first attack, so we have eosinophils and there is one type of cell similar to eosinophil, that is mast cells. Okay, mast cells. Now, these eosinophils has a granules, if you remember, eosinophilic granules, pink color granules, right? So what these granules are composed means, they have histamines. Histamines are very potent, deadliest chemicals. These histamines will destroy the pathogens, okay, by inducing inflammation. By inducing inflammation, histamines will kill the pathogen. That means it is not the eosinophil that is killing pathogen, rather than the histamines within the eosinophil, they are killing the pathogen. That we understood. So how to release these histamine molecules from the eosinophil? When the eosinophil will get activated, this will be activated if the eosinophil binded with a type of antibody called IgE. There are 5 types of antibodies, if you remember. I used to tell that this is a game of destruction. You remember, right? So IgG, IgA, IgM, IgN, IgD. So IgE is responsible for activation of eosinophils, releasing histamines, and histamines will kill the pathogens. Such type of reactions or immune method called as type 1 hypersensitivity reaction. Okay. Then type 2. What happens in type 2 means, here this cell will be binded with something called opsocins. This I will discuss in the next semester. So we have something called complementary proteins. So these C3 molecules will bind to the cells. Okay. Opsocins are the chemicals that will induce phagocytosis. So what happens means, here a macrophage, maybe a neutrophil or a macrophage or even B cell can do phagocytosis. Okay. What is the meaning of phagocytosis? They will engulf the cells or engulf the pathogens. Okay. So how these macrophages migrate towards the pathogen? That pathogen, either it will release PAMs or this pathogen will be binded by a chemical called C3. So with the help of these two signaling molecules, a macrophage will phagocytize the pathogen. Clear? Such type of reactions are called as type 2. Okay, neutrophil C3 mediated phagocytic killing is called as type 2. Okay, then type 3. In type 3, what happens means, in type 3, the pathogen will be binded with some random antibodies. Not random, they are specific. Actually, there are 2 types of antibodies. Listen, this is a new topic to you. Sometimes antibodies, they will just bind to the pathogen, but they won't be specific. Antibodies are not specific, but they will bind to a foreign pathogen. Okay, this binding will red flag the pathogen. Now it is an indication that there is a problem with this particular pathogen. Okay, after binding of these antibodies, this cell will be recognized and this cell will undergo phagocytosis. Other cells will migrate here and they will do phagocytosis. Okay, this is called as type 3 reaction. And type 4, in type 4 reaction what happens is there is something called complement proteins. Complement proteins will bind to the pathogen and they will drill holes. The holes are called as MAC, and this MAC will kill the pathogens. So these are the 4 types of immune reactions we can see. IgE mediated, opsocins mediated, phagocytosis, then antibodies will bind to the cell and that will be phagocytized. So type 3 and type 4 will be complement proteins. Okay, so this is what you understood. Then I will show you a phenomenon here. By the way, students, there are T cell mediated also. Type 4, here T cells will be involved. T cells are very dangerous. They are cytotoxic T cells. Okay, so T cell mediated death is very dangerous. Even normal cells will be died during type 4 reaction. Or simply I will say, patient got a hypersensitivity reaction, maybe he is getting itching. If that is a type 4, he might die. Okay, if that itching is there in the lungs, the patient will die due to suffocation. A severe inflammation happens within the lungs due to T cell activity. T cell will kill normal cells also. That means even the lung tissue will be destroyed due to ongoing infection. Okay. Anyway, type 1, they are IgE mediated. IgE antibodies will bind to the eosinophils. Eosinophils will release histamines. Histamines will kill the pathogen. But there is a problem with histamines. Histamines will induce a severe inflammation. Inflammation is not a good sign. Inflammation will cause, accumulation. The fluid will be accumulated in the tissues. Okay, accumulation of the fluid is not a good sign. Just try to understand, there is excessive fluid within the lungs, so patient cannot breathe. So inflammation will happen in type 1. A severe inflammation will happen in type 1 mediated by IgE. So that is not my issue. So I was just trying to tell the types of reactions. Then I will move forward. Now we have a cell. Just imagine this condition. 30 out of 70 million population. Out of 70 million population, that means out of this Punjab, some 10 patients came as a rare disease. That much rare is our SLE or LE cell. A very rare condition where a patient will have a type of immune problem. That immune problem was their cells. Say for example, this is a neutrophil. Now to this neutrophil, unfortunately this neutrophil cell membrane was damaged. Neutrophil cell membrane damaged and neutrophil nucleus has been exposed to the to the plasma. Now what happens was, the body will start preparing antibodies against the neutrophil antigen, against the nuclear antigen of neutrophil. This is a nuclear antigen and antibodies has been developed against these antigens. Okay, this is one type of antibodies. These antibodies are called as anti-nuclear antibodies. This is nuclear antigen and these are anti-nuclear antibodies. Clear? Now these antibodies can trigger either type 3 reaction or they can also trigger type 2 reaction. These anti-nuclear antibodies will trigger either type 2 reaction or type 3 reaction. Anyway, the cells will be, what happened in type 3? This damaged neutrophil will be engulfed by a macrophage. A macrophage will engulf a neutrophil. Such cells, a cell within a cell, such cells are called as LE cells. LE cells. Lupus cells. Why these are called as lupus cells? First we saw a condition where we saw a lupus reaction or a erythematous reaction where there are red color, red discoloration of the skin with localized inflammation. So this is a normal lupus and I will show a severe form of lupus. So this is a severe lupus condition. So this woman's immune system attacking her own cells. Okay. Now I need to tell one interesting thing. If this neutrophil is nearby kidney, if it is nearby kidney, Now the T cells will start attacking kidney cells also. So kidney failure can happen if these neutrophils are nearby thyroid. Along with the neutrophils, thyroid gland will be destroyed. So wherever the neutrophils are there, wherever this immune attack happening, the surrounding tissue will be damaged. So by that you can understand the consequences. Imagine this reaction in the eyes, patient can go blind. Imagine this reaction in the heart failure, lungs, kidney failure and liver failure. Everything happens within the body. Okay, it's literally their immune system killing their own body. Okay, because of these culprits. Why the body producing antibodies against our own antigens? Why this phenomenon happening? No one knows till today. Till today, no one knows how exactly this reaction going on. Why our body recognizing ourselves as a foreign. Some people saying that this is happening due to UV rays, due to hormonal changes among women. By the way, 90%, 90:10 or 9:1 is the ratio. 9 females and 1 males will be affected by this condition. So UV rays, hormones, other factors leading to these issues. Okay. Yeah. Next one. I will show a few more things. So a severe lupus reaction, another severe lupus reaction, and yeah, one symptom we can see is butterfly-shaped sinus. Okay, a butterfly-shaped sinus is also an indication of lupus. Along with lupus reaction, we need to do CBC, other normal hematological parameters to conclude a lupus reaction, because this reaction also looks like a fungal infection. To rule out the difference between fungal and lupus, we need to do normal CBC protocols, right? Normal hematological test we need to do. Obviously, in the peripheral blood smear, we will identify some lupus cells, LE cells. But listen, these cells are not visualized if the neutrophil is not ruptured. Only a ruptured neutrophil will be engulfed by macrophage, right? So the test will be like this. We will take a test tube. We took the patient sample, patient whole blood, okay? Patient whole blood we took. Then we added glass pieces and we shaped the test tube. By adding glass pieces will destroy the cell membranes. Now the macrophages will do phagocytosis. After 20 minutes or 30 minutes, we will observe the PBS. In the PBS, definitely we must observe some lupus cells. A nucleus, a cell in a cell, that is called lupus cell, right? This cell is also called as TART cell. They are saying TART cells. A cell in a cell is also called as TART cell or lupus cell. This phenomenon is called as lupus phenomenon, LE cell phenomenon, lupus erythematous reaction or phenomenon. And now there are two types of tests to detect this anomaly. One test is directly detection of LE cells, but now they are saying that LE cells is not a gold standard technique because this will mislead the other malaria-like conditions. Okay, malaria also we will see RBC with a parasite, looks like a LE cell. So we have abandoned this test and we have another gold standard test, that is go for detection of anti-nuclear antibodies. Anti-nuclear antibodies test is also there. This is gold standard test. Again there is an issue with ANA. Anti-nuclear antibodies can be seen in other similar conditions like rheumatoid arthritis. By the way, if a patient has rheumatoid arthritis, there is a 90% chance that the patient might develop SLE. Because in LE cell phenomenon, these abnormal antibodies, they will start attacking the other tissues of the body. Other tissues like kidney, liver, everything. Cartilage, bone of the synovial joints. All these things will be affected by or destroyed by these antibodies, leading to further complications in the patient. So this is all about systemic erythematous reaction or systemic lupus erythematous and LE cell phenomenon. And I given a brief notes to you, you can go through it. Understood, right? So in they are saying not only nuclear antigens. These antibodies will target ribosomal antigens, DNA antigens, RNA antigens, multiple antigens are there. Okay, and further complications like a splenomegaly, lymphadenopathy, renal failure, arthritis, hepatomegaly, all organs will be failed by self-attack. That's it. Okay, any doubts in this concept? So there are 5 questions, 2 marks and 3 5 marks questions will come from this topic. Go through it. This is a simple topic. You need not to study the disease in detail, but focus on the lab diagnosis. Okay, if you see disease, that is a very big concept actually. We need not to study the immune aspects of this disease. We just need to see the hematological aspects. That's it. What lab tests are available? What is this phenomenon? What cells you will observe? That's it. Clear? Yeah. Actually, anyway, I will move forward. Next one, you already know Serena Gomez attacked, and there is a scientist called Betty Diamond. Betty Diamond did a very good research on SLE, and she has standardized global standardizations for detection of this disease, and she has also developed some treatment strategies to minimize the complications in these patients. Her name is Betty Diamond, and I will tell there is a small story with this. I also seen this condition when I was studying my second year of BSc MLT, and I was not into it, but I know her, okay, and I will let you know what happened later. Betty Diamond did a good research, and she is from Feinstein Institute from California, okay. Tomorrow I will discuss what happened with her. That's it. That is all about SLE, and tomorrow I will discuss about karyotyping. So before karyotyping, you just go through what is karyotyping, okay? Today go through what is karyotyping. Can anyone say what is karyotyping means? Any idea what is karyotyping means? No. More specific? Any idea? Okay. Karyosomes? Any idea? Eukaryotes, prokaryotes. Now tell me, what is the meaning of karyo means? Nucleus. Okay. What is there in the nucleus? Nucleus has chromosomes. Am I correct? How many chromosomes we have? 46 chromosomes. Counting of 46 chromosomes is called as karyotyping. Counting of 46 chromosomes is called as karyotyping. And we know in some individuals there will be 45 chromosomes, sometimes 47 chromosomes due to non-disjunction euploidic conditions. Down syndrome, one example, a mentally retarded condition where there is one excess 22 chromosome is there. So the overall chromosomal count will be 47. Instead of 46, 47, extra 22 chromosome, and you will be a mentally retarded. So yeah, and there is one condition I will let you know, Philadelphia chromosome. In Philadelphia chromosome, what happens is, I forgot what is the number. So there is a chromosome number 46. From 46, a portion of this chromosome will be binded to chromosome 9. This is chromosome 9, here it will bind. So this is called shuffling of genes. Ideally it should not happen, but unfortunately some of the genes present in 46 migrated to 9. See, genes within the body, just they dislocated, they dislocated, okay, from this location to here. And what happens in the patient, you know, these patients will develop 100% AML, acute myeloid leukemia, that is a cancer. See, one gene present in brain migrated to liver, and now we got a liver cancer. But that is the same genes present in the body. So yeah, that is really interesting, that I will discuss next, okay? How to count these chromosomes, that we'll see. Yeah, so this is all about today's lecture, and I will show you your papers now. Yeah.